So now we move to the sort of the next era, an era again we're currently in, the so-called recombinant DNA era. And again, it was Dr. Hilleman that was the first to take advantage of this by making the next hepatitis B vaccine. So now he was trying to get away from using humans and their blood to make a vaccine. Instead, he wanted to essentially genetically engineer his vaccine, which is certainly an even safer way to make it. And he took advantage of the technology that had been developed by Stanley Cohen and Herbert Boyer, who worked at UCSF and Stanford, respectively, in 1973, to try and express non-bacterial proteins in bacterial cells. This is a picture of Herbert Boyer, and he looks remarkably actually like Jerry Garcia from the Grateful Dead, and this is Stanley Cohen. And what they did was they combined their expertise. On the one hand, one of them was very interested in plasmids. Plasmids are just small circular pieces of DNA which can reproduce themselves in bacterial cells. And then the other was interested in restriction endonucleases. That was Boyer, and he was interested in restriction endonucleases are just proteins that are enzymes that cleave DNA at a particular site. So what using those two things, what you could do is you could cleave DNA. You could then insert a gene, in this case the gene that coded for that hepatitis B protein. You could put it in the plasmid. The plasmid could then infect a cell, either a bacterial cell or a yeast cell, and you could then, when the bacteria reproduced itself, not only would it make bacterial proteins, but it would also make that one protein you were interested in this case, a hepatitis B surface antigen. Actually, the first product, I think, made by this technology, and Boyer formed a company called Genetic Engineering Technology, or Genentech, was insulin. So this was a way to sort of mass produce a single protein in a safe and efficient way. And it led to the first vaccine made using recombinant DNA technology, which was the hepatitis B vaccine. That was the first vaccine to protect a known cause of cancer, in this case, liver cancer. This technology was also used to make a vaccine to prevent another cause of cancer, viral cause of cancer, specifically the human papillomavirus. And again, it was made by taking the gene that codes for the surface protein of human papillomavirus, the so-called L1 protein, by cloning it into, in this case, a yeast plasmid. The plasmid was then infected into really just common baker's yeast, so-called Saccharomyces cerevisiae, and then as the yeast reproduced itself, that protein would actually form a particle that actually looked a lot like the virus, so-called virus-like particle, which could then be purified and used to make a vaccine. In this case, the vaccine prevents the only known cause of cervical cancer, which in the United States accounts for about 10,000 cases and 4,000 deaths a year at least, as well as a series of head and neck and anal and genital cancers in boys as well. So now we move to the next era. It's actually reassortant viruses. There is one vaccine made using this approach. It's actually the rotavirus vaccine, and here's how that vaccine is made. Rotavirus is a virus that causes fever and vomiting and diarrhea in infants and young children. And in the United States, it accounts for about 70,000 hospitalizations and 60 deaths a year prior to that vaccine's introduction in 1960. In the developing world, it's a killer. There's about 2,000 children that die every day from rotavirus infection, about 500,000 children a year. So there's a tremendous amount of interest in making a vaccine to prevent it. And the vaccine strategy used was really different than the vaccine strategies used for everything else. So I'm going to try and explain that to you simply. The rotavirus genome consists of 11 separate segments of double-stranded RNA, and it's each segment codes for a single protein of the virus. It's very easy to remember. One gene segment codes for one protein. If you take the virus and disrupt it and put it on top of a plastic mesh and then run it down through that mesh, what you find is that those genes separate on the basis mostly of size. And so you can see one virus is shown in lane A, another in lane B. If you take those two viruses and infect cells at the same time, what you find is that you can make a virus which actually is a combination of the two, a so-called reassortant virus. So you can see that on the right-hand slide section of the slide, that virus has all its genes from strain A and only one of its genes from strain B. Well, if those two strains differ with regard to their virulence characteristics, meaning what makes you sick, then you can figure out the genetics, in this case, of what makes you sick from rotavirus. In other words, the diarrhea genes, if you will. And also, if those two viruses differ with regard to their capacity to induce a protective immune response, you can figure out the genes associated with making the proteins that induce induce a protective immune response. Now, if those two sets of genes are different, meaning if the genes that make you sick are different from the genes that induce an immune response which are protective, then you can make a reassortant virus which is the best of both worlds, which is to say can reproduce itself in children but not cause disease, but at the same time induce a protective immune response. And that's what at least one of the rotavirus vaccines are. It's a reassortant vaccine. The other rotavirus vaccine is made using the classic means of just taking a human virus and weakening it by serially growing it in non-human cells. So to summarize then this part of the talk, I would say that there are a limited number of strategies to make viral vaccines. You can take a virus and weaken it by passing it in non-human cells. That's the way the measles, mumps, rubella, varicella, or chickenpox, with the oral polio vaccine, which we don't use in this country anymore, but still is used in the world, and at least one of the rotavirus vaccines. You can take a whole virus and kill it, which is the way that the influenza vaccine is made in part, and then in part influenza represents a purification of two surface proteins, but it is essentially a whole-killed product. Rabies vaccine, the hepatitis A vaccine, you can take a virus and use only part of it, which is one protein from the vaccine. It's true with the hepatitis B vaccine and the human papillomavirus vaccine. And then in addition, you can take a virus and reassort it, as is true for the rotavirus vaccine. So that's the way that you would make viral vaccines. Okay, so we've talked about viral vaccines. Now let's spend a little time on bacterial vaccines. Once people could figure out how to grow bacteria in broth, they immediately tried to make vaccines to prevent them. So the way that they would do it is they would grow up the bacteria in broth, and then they would treat it with a series of inactivating agents, things like phenol, otherwise known as carbolic acid, and then they would sell it as vaccines. They called them Bacterins. We had vaccines against Streptococcus pneumoniae or pneumococcus, against group A beta hemolytic strep, which is the kind of strep that causes strep throat, against Neisseria meningitidis, otherwise known as meningococcus, staph, E. coli, Klebsiella, Salmonella. There were a variety of vaccines that were made, and they were called Bacterins. And this is actually an ad for Bacterins. It was made. The ad was made by H.K. Mulford Company, which is in Philadelphia. You see that company ultimately became Merck. These vaccines were very easy to make. They were cheap. They were taken sometimes by mouth, primarily by mouth. And the only problem was that they didn't work. Nor did they have to be shown to work. I mean, this was really at a time in the early 1900s before vaccines were regulated by what is now the Food and Drug Administration, so you could pretty much claim whatever you wanted, and so we had Bacterins. Now, there were a series of key discoveries, actually, that led to vaccines that worked, and I'm going to talk about those now. The first were made by French researchers Roux and Yersin. It's interesting, actually, if you look at sort of the history of vaccines. It starts in Europe and then ultimately comes to the United States, right? I mean, first you had Jenner, who worked in England to make the smallpox vaccine. Then you had Pasteur, who worked in France to make the rabies vaccine. But then, starting with the polio vaccines, really in the 30s and then again in the 50s, vaccine research and development pretty much shifts to the United States. That's true with bacterial vaccines as well, because what happens here is with the first observations made by Roux and Yersin in France were that you could take bacteria like diphtheria, you could grow it up, then you could really remove the bacteria and find that the fluid which was growing, in which the bacteria grew, contained a protein or a toxin that could kill animals. This is a notion then that bacteria make proteins or toxins that then can cause harm. Then Behring, working in Germany, found that low levels of these toxins, if you injected them into antibodies, produced something called antitoxin, what we now know are antibodies directed against those toxins, and that you could take those antibodies and give them to people, and that would be protective. For this, Behring won the Nobel Prize. Actually, when he won the Nobel Prize, he changed his name to von Behring, and he won the Nobel Prize in 1901, which was really that was the inaugural Nobel Prize. And in fact, it led to probably the modern representation of antitoxin is a dog race that occurs in Alaska. Because what happened was in 1925 there was an outbreak of diphtheria in Nenema, Alaska. There was antitoxin that was then flown to Nome. There was then a dog sled that took it from Nome to Nenema, and that dog sled race now is commemorated as the Iditarod Iditarod race, and probably many children know of this because the lead dog's name was Balto, about which a number of cartoons have been made. In fact, if you go to Central Park in New York, you can find a statue to Balto. So I think that's probably many people's conception of antitoxin. And this is again, you can see, it's actually the first, I think, the first of the, I guess, antibacterial agents. In this case, it's not a vaccine in the classic sense. It's not an active vaccine where you're given something and then you make an immune response. Rather, it's a passive vaccine in the sense that you're given antibodies that would have been a product of your own immune response, but is protective. And this was made, again, by H.K. Mulford in 1897. Actually, I think that the first diphtheria antitoxin was available in 1894. But now we move to sort of a more classic vaccine in the 1920s, which is to say you're given something and you make your own immune response to it. And the advance, again, is by Raymond. Again, it's in France in the 1920s, showing that you could take that toxin and inactivate it with formaldehyde and heat. So again, the way to make a vaccine is you take something and inactivate it so it can't cause disease, but don't inactivate it so much so that it can't induce an immune response, which is protective. And that was called antitoxin or anatoxin. Now we know that as toxoid. And the diphtheria vaccine is a toxoid vaccine, which is to say you take the bacteria, you grow it up in broth, you purify that toxin, you inactivate it with agents like formaldehyde, and then you inject it into people and they make an immune response to the toxin, which will protect them against the disease, because diphtheria disease is primarily caused by the toxin. Now you move to the tetanus vaccine. It's actually very much analogous to the diphtheria vaccine. This was made using the exact same technology, which is you grow tetanus bacteria up. Tetanus bacteria makes a toxin called tetanospasmin, and that induces the symptoms of tetanus, which are seen. And tetanus was a problem, especially when Americans entered World War II. This was there were many soldiers who, with wounds that were contaminated with this particular bacterium that causes tetanus. That was a tremendous interest by the American public to try and make a vaccine to prevent this awful disease. And so the tetanus vaccine was introduced in the 1940s, as was the pertussis or whooping cough vaccine. Now this is different. As diphtheria essentially makes one toxin which causes disease, and so the vaccine is just an activated form of that toxin called toxoid. Same is true for tetanus. Pertussis is different. The original pertussis or whooping cough vaccine was made by taking the bacteria, growing it up in a broth, in a nutrient broth, and then purifying the bacteria and killing the whole bacteria. It was a whole bacterial vaccine. It was the only whole bacterial vaccine that we ever used in the United States, and it was introduced actually originally in the 1940s. The problem with that whole bacterial vaccine, it was combined actually with the diphtheria and tetanus vaccine to create DTP, shown here as DTWP because it was the whole pertussis vaccine, but it had side effects which weren't trivial. Children could develop persistent inconsolable crying with a frequency as high as 1 per 100 doses. They would develop high fever, greater than 40.5 degrees centigrade, which is more than 104 degrees Fahrenheit. That would occur in 1 per 330 doses. They would develop something called hypotonic hyporesponsive syndrome, where the child would be sort of floppy and unarousable sometimes for hours, and they would develop seizures, sometimes with or without fever. Now, sometimes although these symptoms were very difficult to watch, they didn't result in permanent harm. So it's not like the vaccine ever caused permanent damage, i.e. a permanent seizure disorder or permanent brain dysfunction. But certainly they were difficult to watch, and that was the side effect of the vaccine. Now, at the time, remember, pertussis would kill about 8,000 children a year in the United States, so it was a price that people were willing to pay until we developed actually a better vaccine. And that's the vaccine we currently use today. Now, because of advances in protein chemistry, advances in protein purification, we're actually able to purify out those proteins of the bacteria that are able to induce a protective immune response, and that there are anywhere from 2 to 5 proteins, which is actually obviously far fewer than the 3,000 proteins roughly that were in the original so-called whole cell vaccine. That fact, the acellular vaccine was introduced in the United States in the early 1990s. Now, the other way to make a bacterial vaccine is to use bacteria that have a polysaccharide coat, a so-called complex sugar. So this is just a picture of that. You can see that that sort of whiter area is just sort of that dense polysaccharide or complex sugar that surrounds bacteria. And there are a number of bacteria that cause disease in children. The pneumococcus, which causes sepsis and pneumonia and meningitis. The meningococcus, obviously, which can cause also primarily sepsis, which is a bloodstream infection, and meningitis, and then the so-called Haemophilus influenza type B, or Hib, is a bacteria also that primarily causes meningitis, but can also cause bacterial arthritis, as well as something called epiglottitis, where the epiglottis, that tissue that sits on top of your windpipe, can swell up and cause suffocation. It can cause pneumonia, that bacteria. So these are bad bacteria, and vaccines were introduced initially just using that, stripping off that complex sugar to make the vaccine, pneumococcus, meningococcus, and H. flu B, in the 40s and in the early and mid-1980s. Now, the problem with polysaccharide vaccines is that they don't work very well in young children, because young children, in order for them to make antibodies, when you ask them to make a response to polysaccharides, that's a so-called T-cell independent B-cell response, which is to say that they make antibodies without the help of a particular kind of immune cell called a T-cell. But babies are very good at making T-cell dependent responses. So the trick was converting the so-called T-cell independent response, which was required with a polysaccharide, to a T-cell dependent response. And that was easily done, actually, by just taking this complex sugar and linking it to a protein. And the proteins that were made are shown here. They're safe, they're inactive, and they were used to make these 3 vaccines that were introduced in the dates that you see here, which now have been remarkable. If you look actually at the Haemophilus influenza type B vaccine, when I trained as a resident, there were 20 to 25,000 cases of Hib every year. We would see a child with meningitis, where I trained at the Children's Hospital in Pittsburgh, almost on a weekly basis, or we would see epiglottitis, or we would see bloodstream infections. But since that vaccine has been introduced, we virtually eliminated the disease in the United States. I would say for physicians, at least my age, this is probably the most powerful of the vaccines, because we saw what happened. Pneumococcal vaccine was introduced in 2000 and has resulted in about a 90% decrease in the incidence of this particular bacteria, and the meningococcus vaccine in 2005. So to summarize then this part of the talk, the bacterial vaccines, like the viral vaccines, are made using a limited number of strategies. Specifically, you can take the toxins made by the vaccine or made by the bacteria and purify them and kill them. It's true for tetanus, diphtheria, and now also pertussis with a so-called acellular vaccine, which is made, the current acellular vaccine is using not only toxins made by the bacteria, but also sort of structural proteins of the bacteria. You can have a polysaccharide vaccine. That's still true for the pneumococcal vaccine, or you can have conjugated polysaccharide vaccines, as is true for Hib, pneumococcus, and meningococcus. Thanks for your attention.